started like celebrating this day since 1987. So since then, every year we have been celebrating this National Science Day across all over the country, in whether it is colleges, universities, or institutes. So just to tell briefly about my organization, I already mentioned that I come from CSIR CMAP. So it's actually one of the 39 frontier laboratories of CSIR. So, and it is one of the unique institute in the world, I would say, because this is the only institute in the world which is dedicatedly working for identifying the importance of medicinal and aromatic plants for human benefit. So it is headquartered at Lucknow, and we have 4 regional centers located in different agroclimatic zones of India. You can see it is like one is in Purara, which is in Himalayas, and then we have Pantnagar, Hyderabad, and I come from CMAP Research Center, Bangalore. So in our center, we are actually working to cater the needs of farmers and entrepreneurs who are interested to take up cultivation, processing, and marketing of medicinal and aromatic plants in the states of Karnataka, Tamil Nadu, and Kerala. So just I hope I know that this group is a diverse group, but I'll try to tell some of the work that we have done in our lab, as well as some of the work which is done by other groups, like how these medicinal and aromatic compounds are made in the plants, and how they can be utilized for the human benefit, and how these molecules can be overproduced in whether it is in plant system or in microbial system. So as you can see here, what is a medicinal plant? Any plant which is capable of producing compounds, phytomolecules, having a medicinal nature is called as a medicinal plant. And aromatic plant is any plant which is able to produce a molecule which is having aromatic or fragrant nature. So I think most of you use some of the aromatic plants in your day-to-day lives, whether it is tulsi or mint. So many plants are there. So in plants, I think some of you are from botany background. So plants have like broadly 2 categories of metabolism. Primary metabolism and secondary metabolism. Primary metabolism is required for the survival of the plant. So without primary metabolism, plant cannot survive. Whereas the secondary metabolites, which are actually the medicinal and aromatic compounds, so they are required for the overall fitness of the plant in terms of its ecological role. So whether it is attracting honeybees or seed dispersers, or even providing the defense against different microbes and insects. So you can see here, these are some of the drugs which are derived from plants. They are used in various applications, whether it is anti-cancer or anti-malarial or even for heart disorders. So it is estimated that around more than 200 medicines which are prescribed all over the world have at least one compound derived from plants. So this signifies the importance of these medicinal and aromatic compounds for the human health. So likewise, we have a lot of products which we use in our day-to-day lives, which use a lot of aromatic essential oils derived from plants. So these, I think, some of the plants, I think all of you can recognize. So anybody can recognize any of the plant here? So they are, at least some of them are very commonly grown. So you can see that there is ashwagandha. Ashwagandha is one of the highly sought after plant in Ayurveda, Indian system of medicine. So it is used in almost all the Ayurvedic formulations because it has such a profound medicinal properties. And likewise, we have periwinkle Kashikanakli, correct? So that is the leaves of this plant produces unique 2 alkaloids which are used in the treatment of various types of cancers. So these are some of the examples I have mentioned here. Likewise, we have other plants, and also in the red I have mentioned the bioactive compounds produced by these plants. You have this Vandelaga is in English it's called as Centella asiatica, and it produces asiaticosides. It has a lot of medicinal value. Likewise, we have aromatic plants. We have davana, and davanol is a very important molecule used in almost all the food flavorings. It is a very unique molecule. So, likewise, we also have tree species which produce, for example, our own sandalwood. So, it produces unique fragrance. So, it is called as sandalols. So, it takes the tree to produce these sandalols 20 years. So, unless the tree matures, it will not produce these important molecules. But it is difficult for like everybody to access this because it is highly priced because of its long duration of the production of these molecules. Likewise, we have different trees which are producing. There is one agarwood. Agarwood, I don't know how many of you know about this tree. Agarwood is a unique tree which is grown in the northeast part of India. And it produces fragrance when the fungus attacks this tree. And this oil derived from this agar wood is the most expensive fragrance so far in the world. It costs about 15 lakhs per kg of oil. So you can imagine how expensive it is to produce this fragrance. So, sorry. So these are the, I don't worry about the complexity of the slide here. So as I mentioned, there are various primary metabolic pathways, and these secondary metabolites are derived from primary metabolic pathways. So, and these are classified as three distinct classes, phenolics, terpenes, and nitrogen or sulfur containing compounds. So terpenes are the largest class. Whatever you see in carrot color, that is a diterpenoid. And then whatever you smell in mint, that is menthol, that is a monoterpenoid. Like that, these are classified based on the chemical nature. And these secondary metabolites, unlike primary metabolites, they are produced in very, very small quantities, depending on the species or tissue or stage or response-specific manner. As I mentioned, sandalwood produces sandalols. No other tree or plant produces sandalols. So it is very species-specific. And also tissue specific. Some are produced only in the flower, some are produced only in the leaf. Likewise, they are also response specific. When there is an insect attack, they start producing, or when there is an injury, they start producing secondary metabolites. So because of this nature, the production of these important molecules becomes very, very cumbersome, and that leads to high price in this market. So this high price and also cumbersome nature can be addressed to a greater extent if we adopt scientific innovations and also like biotechnological applications like metabolic engineering. So we can produce these molecules either in cell culture or at the whole plant level. If you know the entire metabolic pathway, which is nothing but genes involved in the pathway, you can engineer these steps in the microbial platforms, whether it is in E. coli or Baker's yeast, and produce these molecules in a very, very short time as compared to 20 years. If you take the example of agarwood and sandalwood, it takes 10 to 15 years or 20 years to derive those molecules. But if you know the pathway, you can put it in yeast, and in a week's time you can produce these molecules. So that is the power of this scientific intervention. So metabolic engineering, just for the benefit of students, I just want to mention here, it is a term referred as the directed improvement of cellular properties through the modification of specific biochemical steps or more than one biochemical step by the introduction of new ones with the use of course, biotechnology. So there are various steps. I don't want to go into those details here. Various steps where you can engineer the organism to produce the target molecule. So there are different types of engineering. Homologous engineering is nothing but you engineer the host organism itself. So you take the gene and engineer the host organism, whether if it is a sandalwood, you engineer the sandalwood to produce higher oil. So that is called homologous engineering. I am just giving some example here. So this is a mentha. In mentha, there is one molecule called as menthofuran. So menthofuran is not desirable. It is highly allergic. So what scientists wanted to do was they wanted to reduce this menthofuran and increase the yield of menthol. So they blocked this step involved in the production of menthofuran. That way they achieved 50% less of menthofuran, which is undesirable, and they could achieve higher production of menthol. Similarly, I don't know how many of you have heard of Dr. UU2. She won Nobel Prize in 2015. Actually, she was a chemist. She was studying the Chinese literature, like how we have Ayurveda, they have Chinese traditional medicine. So she was reading the literature and she found that this particular plant, that is Artemisia annua, has antimalarial property. But what is the molecule which is contributing this for this antimalarial activity was not known. So she systematically extracted all the compounds and finally identified that this is this sesquiterpene molecule, that is artemisinin, which is responsible for giving the medicinal value in this plant. And this was identified in 1972, but now after several almost 50 years or 45 years, the scientific body recognized her contribution, and in 2015 she was awarded Nobel Prize for this discovery. So similarly, like in this Artemisia annua, so this artemisinin is a very again it is produced in only 1.2% dry weight, so which is very expensive to extract. So what scientists, researchers in UK did was they engineered the pathway by introducing 3 different steps, and they could achieve almost 3 times more artemisinin than the wild type counterpart. So that's how they could reduce the cost of production of this molecule. Similarly, there is also a term called heterologous engineering. Here you take a gene from one organism and put a gene into another organism so that the desired molecule is produced. So here the major example for this is golden rice. I think most of you know that golden rice. Of course, we are not growing it here. Our neighboring Bangladesh is adopted this technology and they are growing. So here golden rice is rich in carotenoids. So the rice endosperm has the entire pathway except one step, that is the phytoin desaturase is not, it's mutated in the rice. So what researchers did was they took daffodil flower phytoin synthase and introduced into rice, and you can see that the white rice it converted into orange, which is rich in similarly they actually went ahead and produced golden rice 2. That was golden rice 1, the first one, and now they have golden rice 2, where they have actually achieved higher production of these carotenoids, which is highly nutritious compared to the normal rice. Similarly, recently Chinese researchers have further went ahead and engineered the pathway where they could achieve production of almost like orange colored rice, which is highly nutritious, having rich carotenoids and the nutritional value. Likewise, tomato also they have engineered. So our own Indian researchers at Delhi, what they did was they took these genes from Artemisia annua and introduced into tobacco. So because Artemisia annua grows only in temperate countries, it is difficult to grow everywhere. So what they did was they took the entire pathway and introduced into tobacco. Tobacco can be grown anywhere, and also the biomass is very high, and easily in 3 months you can get very good biomass. So they introduced this into tobacco, and when they fed the extract, tobacco leaf extract to mice, which is infected with malaria, they could easily cure the disease. So this is another example of heterologous engineering, where you can produce the molecule of interest in a very short time and also for a medicinal purpose. Likewise, people have done a lot of engineering, where you can, instead of lycopene rich tomato, you can have this is a snapdragon flower, where they have taken 2 genes from two genes from this flower and introduced to tomato, and which is rich in secondary metabolites. You can see that old like red tomato has turned into purple tomato. So this is another recent study where they used this synthetic biology, and they used the entire pathway from this one basil, holy basil, and they introduced it to tobacco. And this is the on the right side you see that white powder, which is purified powder from the tobacco. So within a matter of 4 days, they could achieve 3.3 grams of this molecule. So the most successful engineering so far has been in microbes. So I'll just give two examples. Again, I'll give the example of artemisinin. So here they introduced the entire pathway into the yeast, and they could produce almost 40 grams per liter of yeast culture, which takes almost 1 or 2 acres of artemisia growing in the field, and also it takes 1 year to produce this type of this quantity of artemisinin. Likewise, this is another tree, Taxus brevifolia, which produces Taxol, and it is used in the treatment of cancer. And this takes almost like 750 kg of dry bark of this tree to produce 1 milligram of this Taxol, which is very unsustainable and very expensive. So what the scientists did was they took the pathway genes from this tree and they introduced it to E. coli, and you can see that 1 gram per, actually Taxol was achieved like 1 gram per liter of E. coli. So this was a very famous paper published in Science in 2010. So this is the power of synthetic biology, where they could achieve a high production of the secondary molecules. So there are, of course, it's not easy. We have to have a lot of understanding of the pathway. So these are different limitations of the metabolic engineering. I don't want to go into the details here. So again, like to do any metabolic engineering of secondary metabolites, you need a proper platform, whether it is plant cell culture or hairy roots or microbes or even photosynthetic microbes. So I just briefly tell like how like we have used this technology to engineer yeast in our own lab. So I was telling about sandalwood, right? So sandalwood produces, I think all of you would have smelled sandalwood fragrance, and also people use it in all the religious. It is a very sacred recipe for all Hindus, right? They use it for a sacred purpose. So this tree takes about 20 years to produce about 0.3 kg of oil per one tree. So it takes about 20 years and it is very expensive. It costs about 2 to 3 lakhs per kg. So in the long run, it is very unsustainable. You cannot depend on the trees to extract these molecules. So in 2016, a Canadian group, in fact he was my collaborator also, they came up with a pathway. They showed that only 2 genes are involved in this pathway, and we used those genes and we engineered the yeast, baker's yeast. So we introduced the pathway, and you can see that the right side you can see the chromatogram where it shows the yeast is producing almost similar oil profile to that of wild type, natural tree oil. So, and we could show that the simple distillation of this engineered yeast can result in sandalwood oil. You can see that on the right side, a droplet, I don't know whether you can see, there is a droplet. That droplet is nothing but sandalwood oil produced from the yeast. So this is a powerful technology, and we have, of course, filed a patent, and it's going to be awarded soon. So in summary, these metabolic engineering or synthetic biology is a powerful tool where you can use it for production of rare molecules in a very shorter time and in a sustainable manner. So I don't know how many more minutes I have. Do I have 5 more minutes? Okay. So these are some of the work which we have done in our lab to understand the biosynthetic pathway of different plants, and also using that knowledge we are trying to engineer the pathway in plants or even in animals, microbes, so that we can produce these molecules in a larger quantity. So there is a huge global market for these medicinal and aromatic compounds, purified compounds as well as raw herbs. So it is estimated that around 328 billion US dollars in 2023, and is expected to grow almost $500 billion worth of market is there out for these medicinal and aromatic compounds. And it is expected by 2050 is expected to reach around 5 trillion. There is a huge market for these plants as well as for the purified molecules. And also in terms of aromatic essential oils, there is a huge demand. It was estimated around 12 billion US dollars in 2023, and it may reach around 30 billion by 2023. So there are a lot of gaps, of course. So here I have listed out what are the gaps which we need to do in terms of scientific intervention. So we need to develop better high-yielding varieties through classical breeding or through biotechnological approaches. And also we need to develop sustainable and efficient agro practices and processing technologies. Also there is, India is a rich biodiversity country. We have northeast and also we have western Ghats. There is a lot of biodiversity out there, and many of the plants are not explored. So we can explore those new plants with potential phytomolecules and develop agro and processing technology. And also lately, of course, India has invested a lot of research funds in genomics and functional genomics approaches. So of course there is much more need for the investment, so that you can use the latest technology like metabolomics, like ionomics, or different omics technology, so that this technology can be explored for crop improvement. And also we have to adopt latest technologies for pathway elucidation, and also utilize synthetic biology for production of rare and difficult to produce molecules. So well, just quickly, for the benefit of students, I'll take 2 more minutes, if you agree. So there are a lot of opportunities for making career in science, life sciences per se. So I don't know, I think many of you know that there are various fellowships out there for you to attempt and get the fellowship, so that you can have a good research career and try to address various problems which are out there in the society. So there is these are awarded by DBT, SCRP, and also there is a prestigious Prime Minister's Research Fellowship, PMRF. Of course, it is awarded only to some premier institutes like Indian Institute of Science and ICER, but not for others. But other fellowships are there for all of you to explore. Likewise, you have international short-term visitation fellowships for PhD. If you need more details, I can share all this with you people later. And also bilateral student exchange programs, travel grants for attending conferences, where you can go and get connected with the international researchers, so that it can help your research. So of course, after PhD, you can do postdoctoral research fellowship, postdoctoral research also. For that also, Government of India has initiated a lot of fellowship programs where you can avail those facilities. Of course, they are all competitive, but you need to work hard and get those fellowships. If you get those fellowships, of course, it's going to be a very prestigious, and also you can develop your career easily. And also you can go abroad. I went abroad for 4 years, and I came back to serve the nation. So like that, you can go abroad, get the necessary expertise, and come back and work for the nation. These are some of the fellowships which are available. Once you are outside, you can avail these fellowships to come back, and which is almost like associate professor level fellowship for 5 years, and you can establish your own lab and do your research. Once you have necessary expertise, there are many avenues where you can get jobs. Like there are so many institutes in India, like ICMR institutes, DBT institute, ICMR institute, agricultural colleges, premier institutes. So many are there, which are listed here. Of course, this is not comprehensive, but major ones I have listed. So likewise, you have various positions which are there in the industries. Can you imagine there are more than 75,000 food processing startups in India? So which is a very like huge number, and where you have the opportunity to get jobs and also do your research. So these are some of the things which I think I thought it is going to help the students. So with that, I'll again wish you all a happy National Science Day, and I think the discovery of Raman effect by Professor Sir C.V. Raman should inspire all the youngsters, as the theme of this year's National Science Day theme says that you have to empower the Indian youth for global leadership, not for the Indian leadership, for the global level. So once you reach the global level, then only you can help the nation. So it is a collective responsibility for all of us, like institutes and universities, to empower the students to take up science and also nurture scientific temper among all the students, so that the nation is benefited. Thank you, one and all.
At this junction, as a token of appreciation, we would like to felicitate our esteemed Chief Guest, Dr. Dinesh A. Nagegoda. I request all the dignitaries to join and felicitate our distinguished Chief Guest. It's a great pleasure to honor our registrar, Professor Praveen KV sir. I also request FO sir, Dr. Niranjan Raj sir, to accept our token of honor. Now I request Sunita Madam, Director Science School of Sciences, to accept our token of honor. I also request our Chairman, Department of Physics, to accept the token of honor. Starting with Department of Physics, the first prize goes to Shravana NS. Department of Microbiology, first place Atik Anwar, and second place goes to Lavita Flesia. From the Department of Computer Science, first place goes to Karandeep, second place goes to Noori Aisha. From the Department of MCA, first place goes to Pratima, second place goes to Subhash Gowda ES. And from the Department of Biochemistry, first place goes to Soumya B, second place goes to Bhagyalakshmi M. From the Department of Biotechnology, first place goes to Sufyan Nourain, second place goes to Chaitanya HL. From the Department of Botany, first place goes to Anju Naganavar, second place goes to Krutika TB. From the Department of Chemistry, first place goes to Nisarga KJ, second place goes to Raghavendra S. From the Department of Environmental Science, first place goes to Ankit U, second place goes to Akhilesha MS. From the Department of Clinical Nutrition and Dietetics, first place goes to Kaveri Srilavanta, second place goes to Naveen Kumar. From the Department of Food Science and Nutrition, first place goes to Anu SJ, second place goes to Supriya. From the Department of Geography, first place goes to P. Dinesh Kumar, second place goes to Tilak Nandan. From the Department of Information Technology, first place goes to Shanavar Naim. From the Department of Data Science, second place goes to Ramya Radhakrishna. From the Department of Master of Library and Information Science, first place goes to Puneet KR, second place goes to Uday Kumar Gundappa. From the Department of Mathematics, first place goes to Pramila N, second place goes to Shahil Singh. From the Department of Zoology, first place goes to Gaurish Ganapati, second place goes to Nandini KR.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಕರ್ನಾಟಕ ರಾಜ್ಯ ಮುಕ್ತ ವಿಶ್ವವಿದ್ಯಾನಿಲಯದ ಗೌರವಾನ್ವಿತ ಕುಲಪತಿಗಳ ನಮ್ಮ ನೆಚ್ಚಿನ ಕುಲಪತಿಗಳಾದಂತಹ ಪ್ರೊಫೆಸರ್ ಶರಣಪ್ಪ ಬಿ ಹರ್ಸೇಸರ್ ಅವರೇ, ಇವತ್ತಿನ ಮುಖ್ಯ ಅತಿಥಿಗಳು ಹಿರಿಯ ವಿಜ್ಞಾನಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ದಿನೇಶ್ ನಾಗೇಗೌಡರವರೇ, ನಮ್ಮ ಫೈನಾನ್ಸ್ ಆಫೀಸರ್ ಮತ್ತು ಸಿಕಾ ಡೈರೆಕ್ಟರ್ ಆಗಿರ್ತಕ್ಕಂಥ ಡಾಕ್ಟರ್ ನಿರಂಜನ್ ರಾಜರವರೇ, IT ಡೈರೆಕ್ಟರ್ ಡಾಕ್ಟರ್ ಸುನೀತಾ ಅವರೇ, ನಮ್ಮ ಬಯೋಟೆಕ್ನಾಲಜಿ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ನ ಅಧ್ಯಕ್ಷರಾಗಿರ್ತಕ್ಕಂಥ ಡಾಕ್ಟರ್ ನಿರಂಜನ್ ಅವರೇ, ಹಾಗೂ ಈ ಉಪಸ್ಥಿತರಿರ್ತಕ್ಕಂತಹ ಆತ್ಮೀಯ ಬೋಧಕೇತರ ಮತ್ತು ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರೇ, ಮಾಧ್ಯಮ ಮಿತ್ರರೇ, ಇವತ್ತು ದಿನ ವಿಜ್ಞಾನದ ದಿನವಾಗಿ ನಾವು ಹಮ್ಮಿಕೊಂಡಿದ್ದೇವೆ. ಈಗಾಗಲೇ ಇದರ ಸಂಬಂಧ ಪ್ರತಿಜ್ಞಾವಿಧಿಯನ್ನು ಹೋದ ವಾರದಲ್ಲಿ ನಾವೆಲ್ಲ ತೆಗೆದುಕೊಂಡಿದ್ದೀವಿ. ತಮ್ಮೆಲ್ಲರಿಗೂ ಕೂಡ ವಿಜ್ಞಾನ ದಿನದ ಶುಭಾಶಯಗಳು. ಈಗ ತಾನೇ ಅತ್ಯಂತ ಮಹತ್ವಪೂರ್ಣ ಮಾಹಿತಿ ನೀಡಿದಂತಹ ಡಾಕ್ಟರ್ ದಿನೇಶ್ ಅವರು ಹೇಳಿದ ಹಲವಾರು ವಿಚಾರಗಳನ್ನು, ಮಾಹಿತಿ ಪೂರಿತ ವಿಚಾರಗಳನ್ನು ಹೇಳ್ತಾ ಬಂದರು. ಅವೆಲ್ಲವೂ ಕೂಡ ನಾವು ಚಿಕ್ಕದಿನಿಂದ ನಮ್ಮ ಮನೆಗಳಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಸಸ್ಯಗಳ ವಿಚಾರಣೆ, ಅನೇಕ ಔಷಧೀಯ ಸಸ್ಯಗಳು, ಅನೇಕ ಅಡುಗೆಗೆ ಬಳಸ್ತಕ್ಕಂಥ ಸಾಮಗ್ರಿಗಳು, ಮತ್ತು ಪ್ರಸಾದನವಾಗಿ ಬಳಸ್ತಕ್ಕಂಥ ಸ್ಯಾಂಡಲ್ವುಡ್ ಆಯಿಲ್ ಬಗ್ಗೆ ನಾವು ಮೈಸೂರು ಸ್ಯಾಂಡಲ್ ಸೋಪ್ನ ಚಿಕ್ಕ ವಯಸ್ಸಿನಿಂದ ನೋಡಿದ್ದೀವಿ. ಸೊ ಈ ಬಯಾಲಜಿನಲ್ಲಿ ಅನೇಕ ವಿಚಾರಗಳನ್ನ ಈ ರೀತಿ ಎಲ್ಲ ಮಾಡಬಹುದು ಅಂತಕ್ಕಂಥದ್ದು ನಮಗೆ ಒಂದು ಉತ್ತಮ ಪ್ರೇರಣೆ ಆಗಿದೆ. ಇಟ್ಸ್ ಎ ವೆರಿ ಗುಡ್ ಮೋಟಿವೇಶನ್ ಫಾರ್ ಆಲ್ ಆಫ್ ಅಸ್ ಟು ಸಿ ಅವರ್ ingredients, whatever present in our home, but unfortunately we are ignoring a lot of things and we are turning towards westernization instead of modernizing it. That is the irony.